Welcome to the Bioscan Hub channel. And hey, if you find this helpful, don't forget to hit that subscribe button to support us. All right, today we are diving deep into hemoglobin. You know, that powerhouse protein packed inside our red blood cells? We're going to break down the essential lab tests we use to figure out what's going on when things go wrong, how we take a single blood sample and turn it into a clear, life-saving diagnosis. You know, it's such a fascinating paradox, right? On one hand, hemoglobin is absolutely vital. It's the whole reason we can get oxygen to every single cell in our bodies. But on the other hand, if something is off with its structure or how it's made, it can cause some really serious, life-altering diseases. So the big question is, from our perspective in the clinical lab, how do we actually pinpoint what's gone wrong? How can we tell the difference between all the different hemoglobin variants and related conditions out there? Well, that's exactly what this explainer is all about. We're going to walk through the key lab methods that turn those patient samples into definitive answers. So here's our game plan. We'll kick things off with the foundational techniques for analysis. Then, we'll jump into key variants and thalassemia. After that, we'll tackle sickle cell disease, look at some acquired dysfunctions, and wrap things up by looking at the broader markers of cell injury. Let's get into it. Okay, first up, let's lay the groundwork. We need to talk about the absolute cornerstone of investigating these disorders. Because before you can really understand the diseases, you have to understand the main tool we use to find them. And the star of the show, without a doubt, is hemoglobin electrophoresis. The best way to think of it is like a race for proteins. See, we apply an electric field, and different hemoglobins start to move. Because a genetic mutation can swap out an amino acid, it changes the protein's overall electrical charge. This means they move at different speeds, separating out into these neat, distinct bands. It lets us see exactly what's there, and just as critically, what isn't. All right, so now that we know our main tool, let's talk about what we're looking for. It's really important to get this straight. Variants, like what we see in sickle cell, are structural changes to the hemoglobin protein itself. Thalassemias, on the other hand, are production problems. The body just isn't making enough normal hemoglobin. And to spot either one, you first have to know what a normal pattern looks like, inside and out. So this is the typical fingerprint of a healthy adult. The overwhelming majority is hemoglobin A1. That's our main adult hemoglobin. Then you've got this tiny but really important slice of hemoglobin A2 and basically no hemoglobin F, which stands for fetal hemoglobin. This is our baseline, our normal reference point. And this chart just brilliantly shows the dramatic switch that happens right after we're born. A newborn is basically running on fetal hemoglobin, or HbF, up to 90%. But during that first year of life, production flips almost completely to the adult forms. Understanding this transition is absolutely critical, especially in pediatric diagnostics. So, here's the key takeaway. If that switch from fetal to adult hemoglobin doesn't happen the way it's supposed to, that's a major red flag for us in the lab. When we see those HbF levels stay high after 6 months, our diagnostic radar immediately starts pointing towards conditions like thalassemia, where the body just isn't making adult hemoglobin chains properly. And this is exactly why getting an accurate quantity of that HbF is so important. It helps us figure out the severity of the disease. In something like thalassemia minor, you might see just a little bit of persistent HbF. But in thalassemia major, the body is so unable to make normal adult hemoglobin that it keeps cranking out massive amounts of HbF instead. This leads to a really severe and often fatal anemia. Okay, let's switch gears and talk about probably the most famous hemoglobinopathy out there. This one is all about a single infamous structural variant, hemoglobin S. Meet the culprit, hemoglobin S. This altered protein makes red blood cells, which are supposed to be these nice, flexible discs, turn rigid and take on that classic sickle shape, especially when oxygen levels are low. And you can imagine, these deformed cells just can't squeeze through tiny blood vessels, which leads to blockages and the terrible pain of a sickle cell crisis. Now, the first step in the lab is often a simple screening test, something like Sickledex. What we do is create a low oxygen environment for the patient's red cells. If hemoglobin S is there, those cells will literally twist into that sickle shape right on the slide. It's an incredibly accurate screen, but, and this is a big but, you have to remember its limitation. It tells you HbS is present, but it doesn't tell you how much. And this brings up a fundamental principle in laboratory medicine, something we always have to remember. Screening is not the same as diagnosing. A positive screen is just a signal. It's a big flashing light that tells us we need to investigate further with a more definitive test. And in this case, that definitive test is, once again, electrophoresis. And this is why electrophoresis is so essential. It lets us actually measure and quantify the different hemoglobins. This is how we can tell the difference between someone who has the sickle cell trait versus someone who has full-blown sickle cell disease. A person with the trait is a carrier. They make both normal HbA1 and the abnormal HbS. But a person with the disease, they make almost exclusively HbS, which is what causes all the severe clinical symptoms. Okay, so far we've been talking about problems that are written into our DNA, inherited disorders. But now let's shift to a completely different class of problems. Sometimes hemoglobin goes wrong not because of genetics, but because of something we've been exposed to, like drugs or toxins. Here are the three big ones. In every one of these cases, the hemoglobin protein itself is genetically normal, but it's been chemically changed in a way that means it can't carry oxygen anymore. Take Methemoglobinuria, for example. The iron atom in the heme group gets oxidized, flipping it into a state where it's useless for binding oxygen. This can happen from certain drugs or chemicals, or, and this is the most famous example, from carbon monoxide poisoning. And this number right here, this is the truly terrifying chemistry behind carbon monoxide poisoning. Hemoglobin will grab onto a CO molecule over 200 times more tightly than it'll grab an oxygen molecule. What that means is even a tiny amount of CO in the air can completely hijack your body's oxygen delivery system, leading to severe and incredibly rapid oxygen deprivation. When about 20 to 30% of a person's hemoglobin is tied up by CO, the symptoms are things that are so easy to just brush off. A headache, maybe feeling a bit nauseous. This is what makes CO poisoning so insidious. The early warnings are frustratingly subtle. But as that percentage climbs, the situation gets critical fast. By the time you hit 50 to 60% saturation, the brain is so starved for oxygen that the person will lose consciousness. And if it gets above 60%, the outcome is very often fatal. This horrifyingly fast progression from vague symptoms to a life-or-death emergency is exactly why getting a rapid test for carboxyhemoglobin is so absolutely critical in the ER. All right, for our last section, let's zoom out a bit. Diagnosing these disorders isn't just about finding weird hemoglobins. It's also about looking for the collateral damage, the evidence of their consequences, like red blood cells being destroyed or tissues getting damaged. This table shows three other really important tests that help us build the full picture. First, haptoglobin. Its job is to clean up any free hemoglobin that gets released when red cells burst. So if haptoglobin levels are low, it's a huge clue that hemolysis is happening. Then there's myoglobin, which is released from damaged muscle, making it a key early marker for a heart attack. And finally, the PNH test, which directly checks to see if a patient's red cells have a defect that makes them super fragile and likely to break apart. And that brings us to one last thought to leave you with. All the tests we've talked about today, electrophoresis, solubility tests, all of it, they're the classic workhorses of the hematology lab. They measure proteins and what those proteins are doing. But as genetic sequencing gets faster and cheaper every year, how are these two worlds going to interact? Will these protein-based tests stay the gold standard, or will they become more of a backup to a direct genetic diagnosis? That's the evolving world that the next generation of lab professionals will be navigating.